Molecular biology, one principle has guided our understanding of life for decades, the central dogma. First proposed by Francis Crick, it describes the flow of genetic information in a simple, elegant way. DNA makes RNA and RNA makes protein. This one-directional flow became the foundation of modern biology. It explained how genes are expressed, how proteins are made, and how life operates at a molecular level. But as science has progressed, we've learned that biology is rarely that simple. Today, new discoveries are challenging the traditional view of the central dogma, and one of the most fascinating examples is the emerging discussion around the DRT7 protein. So, what makes DRT7 so interesting? To understand that, let's revisit the central dogma briefly. In a typical cell, DNA is transcribed into messenger RNA. This mRNA then travels to ribosomes where it is translated into proteins. Proteins go on to perform essential functions, acting as enzymes, structural components, or signaling molecules. In this model, proteins are the end product. They don't feed information back into the genetic system in a direct, sequence-specific way. But what if that assumption isn't always true? The idea behind DRT7 is that certain proteins may play a more active role in influencing genetic information than we previously thought. Instead of being just end products, they may interact with nucleic acids in ways that affect gene expression, RNA behavior, or even the interpretation of genetic code. This doesn't necessarily break the central dogma, but it certainly stretches its boundaries. For example, we already know of exceptions to the classical model. Reverse transcription allows RNA to be converted back into DNA, a mechanism used by retroviruses. Epigenetic modifications can change gene expression without altering the DNA sequence. RNA editing can modify transcripts after they are produced. Each of these discoveries has added layers of complexity to the original concept. DRT7 fits into this broader theme. It represents the idea that proteins may not be just passive outputs, but active participants in regulating and possibly reshaping molecular information flow. Some hypotheses suggest that proteins like DRT7 could influence RNA stability, guide molecular interactions, or even participate in feedback mechanisms that alter gene expression patterns. If such mechanisms are validated, they could redefine how we think about information flow in biology. Instead of a linear pathway DNA to RNA to protein, we may need to think in terms of dynamic networks where information flows in multiple directions influenced by complex molecular interactions. This has major implications. In drug development, understanding these non-linear pathways could open new therapeutic targets. In genetics, it could change how we interpret mutations and disease mechanisms. And in biotechnology, it could lead to innovative approaches for controlling gene expression. However, it's important to stay grounded. The concept of DRT7 as a challenge to the central dogma is still emerging. It is not yet a universally established paradigm shift, but rather a developing area of research and discussion. Scientific validation, reproducibility, and peer-reviewed evidence are essential before drawing definitive conclusions. That said, science has always evolved by questioning its own assumptions. The central dogma itself has been refined multiple times since it was first proposed. What began as a simple framework has grown into a complex, interconnected model of molecular biology. And that's the beauty of science, it adapts. So where does that leave us? DRT7 is not about rejecting the central dogma, it's about expanding it. It reminds us that biological systems are more dynamic, more interactive, and more sophisticated than our original model suggested. The central dogma describes the flow of information from DNA to RNA to protein. Traditional models treat proteins as end products of this process. Emerging concepts like DRT7 suggest that proteins may also influence upstream molecular events. This challenges the simplicity of the central dogma and highlights the complexity of biological systems. And as research continues, our understanding of life at the molecular level will keep evolving, just as it always has. Because in science, no model is final. Every discovery is just the beginning of a deeper question.
Subscribe to MedPharma Global on YouTube. Get the latest medical and pharmaceutical updates. Click the subscribe button and join our community.